Hello everyone, a very good morning to all of you. So today let's start periodontology. Okay. So the first topic what I'll be talking about in periodontology is basics of inflammation. Okay. See, basics of inflammation is very important such that if you understand the basics of inflammation, the pocket pathogenesis becomes very easy, the periodontitis pathogenesis becomes very easy, and the basic immunological aspects of periodontics becomes extremely easy if you understand the basics. Okay. So with the basics, I'll try to correlate with the surgical part once we move into that. Okay. So let's kick on inflammation. Okay. What is inflammation basically? When a foreign body try to enters the body, our body responds to it. Very basics, right? So when an infection comes, our body try to respond. It tries to attack the enemy, right? So what happens? Imagine this is the epithelium. Okay. So the epithelium, does your epithelium has defense cells? Of course, it has defense cells. Which is the first line of defense? First line of defense is neutrophils. Okay, so the first line of defense is neutrophils. Okay, what is the first line of barrier? First line of barrier will be epithelium. Okay, so understand the difference. So first line of defense is neutrophil, first line of barrier is epithelium. So what happens when a bacteria in the which is present in the plaque and calculus, whatever it might be, gram positive or gram negative, it comes in contact with the epithelium. In this case, junctional epithelium. Okay. So what happens? These bacteria, it will have certain patterns. Say for example, similar to our fingerprints. Every human being has a unique fingerprint, right? So similarly, these bacteria will have a unique pattern so that we can recognize them. Okay. Similarly, our body will have a unique pattern to recognize those bacteria. Okay. So imagine a gram negative bacteria. The most predominantly gram negative bacteria will be aggregative bacteriostomy comitans and P. gingivalis. So let us imagine a bacteria, P. gingivalis. Okay. So this P. gingivalis, as I told you, every bacteria will have a unique pattern, right? So this P. gingivalis will also have a unique pattern. So that is called as pathogen associated molecular pattern. That is called as pathogen associated molecular pattern. Okay. So imagine a P. gingivalis, plaque and calculus present in the plaque and calculus. It comes in contact with the epithelium with the PAMP. Okay. So the PAMP is surrounding it. So what does the body do? It will try to recognize the PAMP. As I told you, similar to PAMP, our epithelium will also have a receptor which is going to recognize this PAMP, right? So imagine this is the receptor. Okay, imagine this is the receptor. This is called as pattern recognition receptor. Pattern recognition receptor. Why? Because it recognizes the PAMP. Okay, so it is nothing but toll-like receptors. So till now the terminologies are clear, right? So every bacteria will have PAMP, pathogen associated molecular pattern. This PAMP will be recognized by a body PRR, pattern recognition receptor, which are nothing but toll-like receptors. We have a lot of pattern recognition receptors like C-type, lectin type, and toll-like receptors. Okay, so predominantly we are going to talk about toll-like receptors. So there are n number of toll-like receptors. So predominantly it will be 2, 4. Okay, so it goes up to 13. Okay, so now what happens? These toll-like receptors, which is present in the epithelium, okay, it will be present in the epithelium. It will be present in dendritic cells. It will be present in macrophages, tissue macrophages. Okay, so we have dendritic cells, tissue macrophages, NK cells, natural killer cells, and a few other different cells in the epithelium. So these different cells, they have toll-like receptors. What is the function of toll-like receptors? It recognizes the PAMP. Okay. Now what happens? This PRR, it catches the PAMP. it has identified the enemy. What it is going to do? What it will do is, this is going to activate a very important pathway called as MYD88 pathway. I don't want to go in depth of this. There is MYD88 dependent pathway, MYD88 independent pathway. It goes through a series of steps. So finally what it is going to do is, it is going to result in stimulation of nuclear factor kappa B. Nuclear factor kappa B. Okay. So what is this nuclear factor kappa B going to do? It is the one that is going to result in stimulation of pro-inflammatory cytokines. What it is going to do? It is going to result in release of pro-inflammatory cytokines. As the name indicates, it is before inflammation. Okay, so the body tries to signal to the blood vessels where most of the leukocytes will be present that some enemy has entered. I'm signaling you, please come and kill the enemy. Okay, so the pro-inflammatory cytokines. What are the pro-inflammatory cytokines? We all know it. Interleukin 1, interleukin 6, TNF alpha, and also it releases chemokine, interleukin 8. Okay, so what is the next step it is going to do? This is going to result in stimulation of the endothelium. Okay, so what it is going to do? It is going to result in stimulation of endothelium, the lining of the blood vessel, because in the blood vessel only we have leukocytes, the defense cells, right? Okay, now once the pro-inflammatory cytokines are triggered, there will be some changes which is taking place in the endothelium. Okay, so what is the changes? The vascular events. Okay, so the first one will be transient vasoconstriction. So the blood vessel will shrink for a very short period of time. Then it's very important, persistent vasodilation. So what do you mean by persistent vasodilation? This blood vessel, imagine if the blood vessel is this much, and when I say persistent vasodilation, it will become this big. It will become this big. So imagine I'm having a pipe of this size and water flows inside the pipe. Suddenly, if I try to increase the size of the pipe, will the water flow speed will get decreased or increased? Obviously, it will go get decreased, because when I am keeping it less, the pressure is going to be more. Okay, when I'm increasing the size, it is going to be less pressure. So what happens? The RBCs will remain in the center. Okay, but the WBCs, they are going to move to the walls of endothelium. Okay, so this is the endothelium. So because of the persistent vasodilation, how this is happening? Because of the release of the pro-inflammatory cytokines, there is persistent vasodilation. So that is going to result in migration of these leukocytes. I'm marking leukocytes as L. So it moves towards the vessel wall, that is the endothelium. This is called as migration. Okay, so because of these chases of the blood, the leukocytes migrate to the walls of the endothelium, and there are certain receptors in the walls of the endothelium which will be activated by these pro-inflammatory cytokines. We have what is called as selectins, P selectins, E selectins, L selectins. Okay, P, E and L selectins will be there. And apart from that, we also have what is called as addition molecules, ICAM, intracellular addition molecules. So these P, E, L, ICAM will be activated, which means it will be expressed. So normally it will not be expressed yet. Now because of these pro-inflammatory cytokines, two things are happening. One is the transient vasoconstriction, which is followed by the persistent vasodilation. Because of the increase in the size of the endothelium, the blood vessel flow is going to get reduced. So the RBCs stay in the center, but the leukocytes migrate to the walls of the endothelium. In the endothelium, certain receptors or certain cells are activated, the P, E and L selectins and ICAM. So what happens? The leukocytes have migrated to the wall now. Then what happens? It rolls over the selectins, P, E and L selectins. So this is called as rolling over. This is temporary addition. Okay, so it is not going to result in permanent addition of the leukocytes to the vessel wall. It will going to cause temporary addition. So once these leukocytes migrate to ICAM, intracellular addition molecules, that is going to result in permanent addition. Okay, so once it is attached to this ICAM, what it is going to do is it is going to bind strongly to the walls of the endothelium, and the next step will be increased vascular permeability. Okay, so persistent vasodilation causes the vessel wall size to increase, and the leukocytes migrate, rolls over, gets a firm addition with ICAM. And because of the increased vascular permeability, okay, there's going to be numerous pores in the endothelium. Okay, the pores are going to result in the expulsion or the movement of these leukocytes outside the blood vessel. Okay, so another one important point is we have ICAM in the endothelium. So something should be present over the leukocyte to bind to the ICAM. That is called as integrins. Okay, so the endothelium will have PEL selectins ICAM. The leukocytes will have integrins. Only when the integrins that which is present in the leukocytes bind to the ICAM, there is going to be permanent addition. Only when there is permanent addition, the leukocytes moves outside. So this movement of the leukocyte outside the process is called as diapedesis. Okay, so through the pores of the endothelium, how the pores are created? Because of the release of the pro-inflammatory cytokines, there is going to be increased vascular permeability. Very simple, right? Okay, now I told you a very important thing called chemokine, interleukin 8, right? So this chemokine, it is going to send signals once these leukocytes have migrated outside. It is going to take this leukocyte, okay, to the area where the bacteria is present. I'm just drawing this bacteria to here, okay, to the area where the bacteria is present, and this is going to result in phagocytosis. So phagocytosis means there is oxygen dependent, and there are a lot of ways bacteria is going to the neutrophil is going to kill the bacteria by the release of the reactive oxygen species. Okay, so what happens, the neutrophils migrate to the area of the bacteria. It is going to engulf the bacteria, it is going to release the reactive oxygen species, and it is going to kill the bacteria. Okay, now imagine that this is a neutrophil, it has killed the bacteria. Okay, what it will do is it will express these bacteria, as fragments. Okay, so these fragments which will be expressed by the neutrophils, or in simple terms, the bacteria which is expressed by the neutrophils will be taken up by the dendritic cells. Okay, so the dendritic cells express these fragments, dendritic cells. So sometimes this process will be skipped. That is, the neutrophils will engulf the bacteria will be killed into fragments. Directly, the dendritic cells will take up these fragments and it will express on the surface of the cell. Okay, so this is called as MHC2 complex 1 and 2. So dendritic cells will take up the fragments of the bacteria which are phagocytosed by the neutrophils. It will expressed on its outer surface. So via what are the two things? MHC1 and MHC2 receptors. Major histocompatibility complex 1 and major histocompatibility complex 2. So via these two complexes, the fragments of the bacteria are expressed by the dendritic cells. So till this phase, it is called as innate immunity. Okay, so till this process, it is called as innate immunity. Bacteria identified by the host PRR, it triggers the release of the pro-inflammatory cytokines, which is going to result in certain vascular events of inflammation, persistent vasoconstriction, followed by transient vasoconstriction, followed by persistent vasodilation, followed by increased vascular permeability. That is going to result in migration of these leukocytes to the area of the bacteria, which is phagocytosed by the neutrophils, and it is expressed by the dendritic cells via MHC1 and MHC2. Till this very clear, right? Okay. Now what happens? The expression of these fragments, simple terms antigens, by the dendritic cells results in activation of adaptive immune response. So this will be taken up by the T lymphocytes. So we have two types, we all know that. T helper and cytotoxic T cells. Okay, so the T helper cells, it is going to stimulate B cells. And what is the B cells going to do? It is going to release plasma cells and memory cells. So these plasma cells, they are nothing but immunoglobulins. IgG, IgM. And memory cells, they are going to fight against the upcoming infection. Okay, more fast. And cytotoxic T cells, they are going to kill the bacteria. So this is how an innate immunity is linked to the adaptive immunity by the dendritic cells. Okay, so the T lymphocytes, T helper, cytotoxic T cells. T helper activates B cells, produce plasma cells, produce immunoglobulins, and cytotoxic T cells is going to kill the bacteria. So now what is going to happen is, if there is a bacterial load is less, the neutrophils will phagocytose, the process is going to get over. But if there is increased bacterial load, what is going to do is, these immunoglobulins which are released during the initial attacks or initial phagocytosis of the bacteria, which is present here, they will move to the site, okay, of the bacteria, and they will opsonize the bacteria. Opsonization of the bacteria is going to take place, and also it will activate complement system. Okay, C3A, C3B, all those things. So once a bacterial load is more, these immunoglobulins, especially IgG and IgM, they are going to move to the site, they are going to stimulate complement, they are going to opsonize the bacteria, increase the speed of the phagocytosis. Okay, this is how the process of inflammation takes place. Okay, so always remember, the bacteria, even if it does not have that much amount of virulence factors, it will be very difficult to kill a bacteria. Okay, but imagine P. gingivalis and Aggregatibacter actinomycetum comitans. I'll talk about that in the upcoming videos. It will have a lot of virulence factors. It will deactivate neutrophils. It will deactivate neutrophil chemotaxis. It will inhibit phagocytosis. So it does all these kind of things, which makes it even more difficult. Okay, that is why P. gingivalis and Aggregatibacter actinomycetum comitans are the very key important pathogens that is causing periodontitis. So now, if you know this pathway of inflammation, the periodontitis becomes very simple. Now I'm going to tell you another one important thing. As I told you in the first phase, that is the innate immunity phase, there will be release of pro-inflammatory cytokines, right? Okay. And in addition to the release of the pro-inflammatory cytokines, there is going to be release of prostaglandins. So these prostaglandins, they increase the expression of rank and rankle. So what is rank and rankle? Rankle will be present on osteoblast, rank will be present on osteoclast, cell surface receptors. Okay, so these two are cell surface receptors, osteoblast and osteoclast. So only when this is expressed, what is going to happen? There is going to be bone resorption. So normally it is in unexpressed state. So when it is expressed, now you tell me, that is the reason when the patient is having plaque and calculus, there is going to be trigger of the pro-inflammatory cytokines, release of prostaglandins. That is why the patient is having bone loss. Is it clear now? Is it linking? Okay. So see, like this, I will link each and every things in perio with the basics of inflammation. Okay. So I will try to add few things into this basics of inflammation, and I will try to correlate that with the diseases. So before moving into the perio, let's get the scratch out of it of inflammation. And I want you all to go through this again, and I want you all to make yourself thorough in this inflammation, so that in the upcoming videos, whatever I take, I will link to this inflammation. Okay, so patient comes, bone loss, inflammation. Systemic diseases, diabetes, increased pro-inflammatory cytokines, periodontitis. Heart patient, atherosclerotic plaque, increased pro-inflammatory cytokines, bone resorption. Link of perio, cardiovascular, diabetes, everything revolves around these pro-inflammatory cytokines. Okay, so what is now you are going to tell me why it is released and how it is released when I talk about each bacteria. Okay, so when I talk about P. gingivalis, if I tell you it inhibits neutrophil chemotaxis, then immediately you should remember, okay, neutrophil chemotaxis, if it is impaired, then there is no going to be phagocytosis. Okay, so I hope this video makes sense. This is about the basics of inflammation in perio. In the upcoming slides or upcoming video, I'll be talking about the other important chapters. Thank you so much. Have a nice